All right, so this presentation is to talk a little bit about how plants respond to caterpillars and aphids and how they're different and same, and then what happens when they're both feeding simultaneously. And this has a lot to do with the plant defense hormones. Again, these are very ubiquitous and ancient in regards to being present in plants and responding. So ancient herbivores and plants were battling it out, and these hormones, plant defense hormones, have been present across the animal kingdom for all practical purposes as best as we know. And so aphids and caterpillars are dramatically different in regards to how they feed on plants. Caterpillars, as you know, have big mandibles in comparison to aphids, and they'll literally chew through plant material, tearing at it and eating it, and then they tend to spit on it a little bit, regurgitate on it a little bit. While aphids have a straw for mouth parts, so they're not chewing insects. What they're called is piercing, sucking insects. So they have a straw-like mouth. And depending, I believe the mouth parts will go into the phloem. If you're not familiar with plant vessels, there's xylem and phloem tends to be the sugary solutions that are made from photosynthesis. And depending on what type of piercing sucking insect, some will get into the xylem, which is the more water that comes up from the roots, and then some get into the phloem. And what they'll do is they'll actually put their mouth, they'll come up and they'll put their mouth into a plant, get between, I don't know if they actually get between the cells, but they'll stick their straw-like mouth part between, like my fingers going, whenever I'll see that, but basically it's a straw-like mouth part that's kind of, some vary on the degree of sharpness, but the point is, once the mouth part is in the plant, they'll actually release salivary factors that harden around it to make like a straw. So they have these piercing, sucking mouth parts, and then they can actually form a hardened shell around their mouth parts, kind of protecting their mouth parts, and forming a straw-like structure. And so these are aphids on the left. These are the potato aphids, so they're specialists of solanaceous plants. And they often give live birth and are pretty much ready to asexually and sexually reproduce. Aphids tend to be wingless until the plant starts to become a poor food source. Then the aphid can actually form wings in some cases. There's a triggering mechanism that'll allow the next generation or so of aphids to have wings so they can fly off to another plant. So that's pretty remarkable. Over here we have our caterpillar. It's another generalist that's pretty closely related to Helicoverpazea, the caterpillar we often talk about. In this case, it's the beet armyworm. It's Spidoptera exugia, and again, it's a generalist. It also has the salivary enzyme glucose oxidase present in its saliva. This is a pretty serious pest across the world. When I say pretty, I mean it is a serious pest across the world. And it's kissing cousins as well. They're all kind of part of that family, Noctuidae.
Did you say that the aphids were specialists or generalists?
These are specialists on solanaceous plants, meaning tomatoes, potatoes, tobacco, things like that. And so there's lots of different types of aphids. You might have a soybean aphid, for instance, that'd be specialist on soybean, or specialized in soybean. And again, they're piercing, sucking mouth parts. So feeding by chewing caterpillars causes severe damage to plant tissues. So in addition to they have factors in their saliva, as you're starting to get. Some of them may suppress some of the plant defenses like glucose oxidase in some cases, depending on the plant. But in other cases, there's these what they call fatty acid amino acid conjugates. Basically, it's a fatty acid type spit factor that's found in the caterpillar's regurgitant or spit that triggers plant defenses. So it's not just the chewing that triggers plant defenses, but it's also the regurgitant as well. And they tend to turn on the jasmonate defenses. So going back with this, so mechanical wounding and elicitors in the caterpillar's oral secretions activates the jasmonate pathway, leading to all these anti-nutritive defenses like protease inhibitors, polyphenol oxidase. These are defenses that, again, make it difficult for the caterpillar to digest the protein that is present in the plant tissues. Now, aphids, on the other hand, are kind of thought of as gentle feeders. Again, they're piercing, sucking mouth parts. And again, as I mentioned, full feeders as they're getting nectar, and it's just going through the body. They're just sucking up the solution as the plant is made. So there's somewhat of a carbon nutrient sink in the plant. So they're taking in more of the nutrients in that localized area. So the leaf can hold the aphids there for a long time while the caterpillars chewed up and destroyed the leaf and has moved on to another portion of the plant. The other thing, though, to keep in mind is aphids bring diseases. So aphids can get viruses, plant viruses, and the plant virus will actually develop inside the aphid, go through replication and go through several cycles. And then the aphid can go to another plant and infect that plant with a plant virus. So the plant virus actually infects the aphid and the plant in many cases. So you got to think that the plant at least knows, in quotes, again, that's just a nice euphemism, just to make it easier for us to understand, but at least I think many plants will recognize if an aphid's feeding that it needs to kind of gear its defenses in part towards plant viruses. That would be a good hypothesis to presume. So again, they act as sinks, taking in nutrients, and they tend to stimulate a different set of defenses like peroxidases and lipases. These may be effective against the aphid, but they also might provide protection against viruses. So the pectinases tend to make the plant cell walls stronger. And peroxidases do that. So they make the plant cell walls stronger. So the aphid feeding could actually make it more difficult for other aphids to feed on it by this, through making the cell wall stronger over time. So again, you've seen this pathways before. We have their induced resistance pathway and our systemic-acquired resistance pathway. We tend to think of the systemic-acquired resistance pathway as the salicylic acid pathway, and the induced resistance pathway is primarily the jasmonate pathway. And so if a caterpillar is feeding, we tend to stimulate these defenses, and in tomato plants, we'll actually stimulate cysteamine, which is a polypeptide. But in all these cases, we'll end up stimulating those anti-nutritive defenses, again, protease inhibitors, polyphenol oxidase, threonine deaminase. These are all examples of plant defenses that, in a sense, give the caterpillar a stomachache, in quotes. While, again, the systemic-acquired resistance pathway, the aphids tend to stimulate hydrogen peroxide, which will then downstream stimulate salicylic acid and a variety of plant defenses, plant pathogen defenses primarily. But again, they may have some effect against the aphid. Now, as I've told you before, there's crosstalk between these two different pathways. So if the caterpillar is feeding on it, then it may suppress salicylic acid responses if the aphid comes along later, and vice versa. So in other words, if the aphid's been feeding and the caterpillar is feeding, what would happen to these defenses? Would they be stronger or weaker? obviously if there's crosstalk and there's potential to co-suppress each other, the hypothesis would be that they'd probably be lower because salicylic acid would downregulate jasmonate, and then ultimately that would downregulate these protease inhibitors. But anyway, there's multiple outcomes, and so you should think about all the possible outcomes. You could call these hypotheses of sorts. But if we have these two herbivores feeding at the same time on tomato plants, there could be an additive response. Maybe the plant defenses like protease inhibitors that the caterpillar normally stimulates will increase additively together. Maybe so if a caterpillar feeds alone, it's here, it's maybe several, a little bit higher, and then if aphid and the caterpillar feed, it's even higher, right? So an additive effect. Or maybe there's no trade-off. Maybe when the caterpillar and the plant and the aphid feed on the plant stimulates their respective defenses irrespective of these plant defense pathways, and so there's no difference. If the aphid's feeding or the caterpillar's feeding, whatever the plant's going to do for the caterpillar and the aphid, it just does. It's going to have the same amount of plant defenses as if they were just feeding by themselves. Or perhaps there is a trade-off. Maybe these two different hormone pathways interact with each other, so that if the caterpillar's feeding on the plant and normally would raise the jasmonate and the plant defenses a certain level, maybe then it would have to come back down a little bit if the aphid's there, because the aphids are suppressing the caterpillar's defenses through those two pathways. That's the hypothesis we would have. So there would be a trade-off, is our argument, because the plant can't handle these two pest problems equally well. So it's going to have to focus its energy towards one or the other. And again, we think there's going to be a trade-off because of what we've seen in the literature, which shows that there's a co-suppression of the jasmonate salicylic acid pathways. So here's our protocol. We had leaves on tomato leaves. So we had bags on tomato leaves. And in one bag, we would have aphids. In another bag, we would have the caterpillar. So we'd bag the leaves. Or in the control, we would bag it, but not put anything on it. We tried to use around 40 adult aphids. The reason for that is we were trying to approximate the amount. Aphids are a lot smaller than caterpillars, so we wanted to approximate the amount of mass, insect mass, that was feeding on the plant. So 40 aphids we were considering equivalent to having a third instar caterpillar on it. Again, this was an assumption on our part. So we have, again, randomly assigned these treatments. In good experimental design, you randomly assign. Some of the plants, 1/4 of the plants would be control plants with just the bags on them. The other treatment would be plants damaged by just the caterpillar. Third treatment would be, and this would be another 25%, would just be damaged by aphids. And then on our fourth treatment, we would have either aphids and caterpillars feeding. Now these are on alternative leaves, so they're not on the same leaf, so we don't have aphids interacting directly with the caterpillars. But what we did do is choose leaves that we know share a common veinage, so their signaling could kind of come in contact with each other. So it's just, if you think about a rule on a plant, we know which leaves kind of are part of the same xylem and phloem pathways, and so that's how we kind of chose it. Then after 3 days, the leaves were either analyzed through gene expression, which would be our microarray analysis, which you heard about a little bit last week, or we would do chemical analysis, which means we would measure the protease inhibitors, and possibly polyphenol oxidase and other chemicals. And then we would also do a biological assay. Now remember, a biological assay is when we actually use the insect as our assay. So we measure the amount of growth occurring in the caterpillar, for instance. So here's our microarray slide. This tells you that after 72 hours, we collected the leaves. We purified the RNA. We then purified the total or messenger RNA from the total RNA. And then through reverse transcription, we made cDNA that either contained the site 3 or site 5. And as you remember from the last presentation last week on Intro to Microarrays, we talked about how these different spots are either yellow or green and so forth. And so that tells us which genes are being turned on. So again, if you've forgotten a little bit about how microarrays work, I would refer to you to our Intro to Microarray slide or talk that happened last week. So again, we're able to now look at different spots and say, oh, this one's being turned on by the caterpillar, this one's being turned on by the aphids, or maybe this one's being altered by the caterpillar aphid treatment. So that's what this allows us to do. And again, relative amounts, full difference.
I have a question.
Yeah.
Okay, so if the aphids are feeding on the plant and it's moving the nutrients around because they're feeding on the phloem, right? Wouldn't that still induce the IR pathway for the plant because it's like herbivory happening?
Well, apparently it doesn't do it into the way that the chewing insect does. So even a virus will take up some of that nutrients, but the fact that that strong damage didn't occur where we have bits of, so a lot of that jasmine pathway is turned on because bits of that leaf have been damaged. And so there's actually wound factors in the leaves themselves that are being bypassed by the aphids. So it's the fragments of the cell wall that aren't really, they're helping trigger some of that jasmonate pathway. And then it's heightened by the caterpillar spit. And the aphids are so gentle with it that this doesn't cause that kind of damage enough to trigger that response. But it does turn on the salicylic acid pathway, so it does have a response. But I'm not, again, it might be because the plant's responding to the potential virus infection that could be coming, that it's tuned its evolution towards dealing with the aphid as a vector more than as a herbivore, is a kind of a speculation. But either way, experimentally, we've seen that aphids and whiteflies don't trigger the jasmonate pathway. But again, there's always going to be exceptions and things like that. We were able to get these tomato microarray chips from the Boyce Thompson Institute. And things were still pretty new when we got into this. There wasn't a lot of microarray studies at this point with plants and insects. So we were still pretty new kids on the block trying to figure things out. But remember that blue box that's in my lab that I showed a picture of? This is the microarray scanner that we used. And we had to figure out all the statistics and stuff like that. We also used an azocasine method, excuse me, to measure protease inhibitors. So the tomato plant's going to make a protease inhibitor that we can measure in the wounded plant versus the control plant. Now, azocasine is, I don't know if you're familiar with, you should be maybe familiar with casein. That's actually a milk protein. The azo is the dye that's been added to it through some company like Sigma that Ben Boda would have worked at in his early days. So we would have bought azocasine from probably the Sigma chemical company. And what happens is the proteases will digest the casein, and that'll liberate this azo group to be analyzed with a spectrophotometer. So that gives us an idea of how much of this milk protein is digested by proteases. Now we'll mix that in with the leaf material, excuse me, the leaf material that's been come from these two different treatments. And we'll see less digestion because of the protease inhibitors. So this is all chemical enzyme assays that we're talking about. So our enzyme assay allows us to measure the amount of protease inhibitors. And again, that's just telling you in a nutshell. We also, looks like he did if I remember right, because some parts of the test, most of the research I did was the molecular portion, and a fellow named Cesar Rodriguez did the bioassays and the chemical assays. So you'll get to see whether or not it affects growth. And I don't know if he did preference. I don't think so, but let me double check as we go through. So anyway, this is the results. This is just a small percentage of it. So we probably had thousands of genes that were altered. And then I went through and picked out genes that were specifically related to plant defenses that we knew about. For example, aminopeptidase is a plant defense. It's on the top row here. And this is all relative to each other. So we use the non-wounded treatment as our control treatment. And then we look at the gene expression in relationship to the non-wounded. So what this means is when the caterpillar fed on it, it had a 4.4-fold higher level of transcripts, or at least as far as the microarray is concerned, it was brighter than the other two treatments. So this means, again, we're saying we had a relatively 4 times as much of this aminopeptidase for the caterpillar treatment than the non-wounded treatment. But you can see it's actually dramatically higher than the aphid treatment. And then look what, this is kind of crazy, the aphid and caterpillar treatment bring it back down. So maybe there is some trade-offs. Let's look at some other genes. Here's arginase, another plant defense. If you haven't, I don't know if you've read the paper yet, but arginase is important for breaking down arginine. Again, it's highly stimulated by the caterpillar. The aphid doesn't stimulate it. But when the aphid and the caterpillar are both on there, it comes down a little bit. Now, since they have the same letter, that means there's no significant difference, but there is definitely a trend there. And then in this one, there was a significant difference. We also see this with protease inhibitors. Look, 16-fold higher. Now, this kind of goes along with what maybe Morgan was asking. There was still a little bit of an increase in these protease inhibitors by the aphid feeding, kind of like you were asking about. Like, look at this one, protease inhibitor 2. It was higher, maybe not significant because of the letter, but look at this caterpillar cost 40-fold increase essentially. And then again, the caterpillars and aphids brought it down somewhat. So we see this trend over and over again. And so we actually did QPCR and saw the same results. Here's caterpillars really stimulate arginase, and caterpillars and aphids bring it back down. The aphids aren't as high. And so see, it's similar to what we saw in the microarray. Same thing here. Another plant defense, thurine deaminase, is really stimulated by caterpillars, but aphids and caterpillars bring it back down, and aphids don't stimulate that much. So there does appear to be a trade-off. If you look at their body weights, this is a little strange. Let me pause it for a second. Oops. All right, can everybody see I think everybody can still see the slide, right? So, thank you. And so this one's a little different. This is looking at larval weight. And so this to make sure you understand what's going on in this picture. If we collect leaves and feed it to caterpillars to look at their larval weight of the caterpillar, if the caterpillar feeds on a control treatment, this is its body weight. Looks like it's around 0.4 milligrams. I think it means 400 milligrams. That would make more sense. And if a caterpillar, if you put caterpillars onto plants that are fed on by caterpillars, look what happens to their body weights. It was dramatically reduced. Now, if you put caterpillars on plants that were previously fed on by aphids, the body weight goes up. So I think that's really interesting right there, but we're going to look at one more thing. So again, if the caterpillars were if the plants were previously fed on by aphids, and then you feed it to caterpillars, their body weight is actually increased. So the plant is actually more susceptible to the caterpillar after the aphids fed on it. The caterpillars grow better on it. But here's something kind of interesting down the other end that wasn't a trade-off. This actually looks kind of additive. If you have caterpillars and aphids feeding on, and you put the larvae on it, their body weights actually do worse than if they're fed on caterpillars alone. Now, granted, this is a trend. There's no significant difference. These are significantly different than those, though. So it seems like caterpillars are driving this response mostly. But it doesn't seem like there's much of a trade-off in regards to these plant defenses. In fact, mortality is actually a little bit higher if you put caterpillars on plants that are previously fed on by aphids and caterpillars. While if you put caterpillars on plants fed on by aphids, mortality is less. So you can see if the aphids are there before the caterpillars are there, it's a pretty big benefit to the caterpillar. The plant has invested a lot of energy gearing up its defenses against aphids, and so it's very susceptible, it seems like, to the caterpillar. But it doesn't seem like much of a trade-off if both of them feed on there at the same time. Now, this seems a little contradictory to what we were seeing in the QPCR results, because you can see the plant defenses, there is some trade-off. But again, this is still higher than the aphids alone, so those defenses are still important. And they did come down a little bit because of the aphid feeding. But there might be other factors too. Maybe the aphids are functioning as a sink a little bit, or maybe there's just enough there, it doesn't really matter between these two. So again, molecularly, there is a trade-off, but at least in regards to this experiment, there's definitely a trade-off molecularly, but when you talk about the bioassays, the caterpillars do horribly when feeding on plants previously fed on by caterpillars or plants fed on by aphids and caterpillars, in comparison to caterpillars feeding on plants fed on by aphids by themselves. We see this trade-off again with polyphenol oxidase. This also binds up plant proteins, so they're no longer edible. And so that's important to note. So again, there was a trade-off, but this is still quite a bit higher than aphids alone. But what you're seeing though is that aphid feeding does have an effect on the signaling of caterpillar defenses. We see this time and time again, this repeated. If aphids and caterpillars are feeding on jasmonate defenses, and that's important to remember, jasmonate defenses seem to come down. But the actual bioassays and some of the enzyme assays don't seem to necessarily reflect what we saw with the molecular data. This may be for a variety of reasons. One, we didn't measure all the different types of protease inhibitors. We just kind of broadly measured them. So again, the trade-off, in molecularly speaking, happens between the caterpillars and aphids, but not on the bioassay level. But there would probably be a trade-off if it's just the aphids feeding first. So again, it seems like when you're talking about gene expression, the tomato plants appear to be stimulated in a manner that you would expect by the respective herbivores. In other words, the caterpillars really trigger those jasmonate defenses strongly. Aphids not nearly as much. Caterpillars reduce photosynthesis more than aphids do, that's actually pretty closely related to jasmonate, by the way. Dr. Mishak Aphid, he's our plant physiologist, has studied jasmonate back in the day, and has shown a lot of that has to do with photosynthesis on some of the work he's done, along with lots of other people, of course. So tomato plant gene expression responds to simultaneous herbivory differently than single herbivores. So, again, we see this change in these jasmonate defenses coming back down. These were some of the researchers involved. This is Cesar Rodriguez, well, not just involved, these are co-authors. Cesar Rodriguez is a professor at Rutgers University. The focus of his research now is on blueberries. So he does research. I believe he's a full professor now, and a certain percentage of his duties are research, and some of his duties are extension, and he probably has some teaching duties along with working at this lab. This is Heiko Vogel. He is a scientist at the Max Planck Institute in Germany, and I'd love to get out there again and visit him at least one more time and do some research out there. And then this is Spencer Williams. He was an undergrad student with me when I first came to Western and was involved with one of those high school students back in the day. So I took him out to Germany with me when we did these research projects. And then this is Jennifer Thaler, who's also, all these people are very famous scientists outside of this guy. Jennifer Thaler is really well known in plant herbivore field. She works at Cornell University. So I think one thing kind of cool that you might get out of this, here's Rich Musser at Western, good teaching university, but not as famous. I think we can all agree it's not as famous as Cornell or Rutgers University or the Max Planck Institute in Germany. Yet I was able to create all these collaborations. So you can do, like I said, don't limit yourself, your imagination, your ability to reach out to people. And I could be doing research with all these people at this now still. So again, it's all available if you're motivated enough. So if you end up getting a PhD and finding yourself in a smaller university, your teaching level is going to be higher, but you can still make things happen if you're motivated enough. Obviously, it's a tiring day. It's a long day, and that's true whether you go to Rutgers University or Cornell. These are all people that have devoted their lives to it. So anyway, I want you to at least be inspired to realize that like you said, even if you see from my students, they've all had some pretty good success stories. So some people get caught up on names, of schools, but you realize you can have a great career at a lot of universities. Anyway, does anybody have any questions? All right, well, I thought it was a pretty good day. And even though I realize, obviously, I don't know what date I gave you for any of this stuff, but realize there's no rush on getting back with the Ben Boda review or anything like that. Just get it when you can. That's true for all the assignments right now. And otherwise, I think we're done for tonight. Sound good? So you're welcome to go ahead and sign out. I hope you enjoyed the presentations and have a good rest of the week.
Bye, Dr. Musser.
Take care.
Thank you, you too.
Thank
Good
you.
night.
Good night.